Well, I think we're getting really close. At this meeting, data were presented by a company named Avidity Biosciences. And this company has a drug which can go specifically to muscles and that will block the messenger RNA of DUX4 to prevent the toxic protein production. And they presented their results for a 1-year follow-up of a small number of patients with that drug at different doses. And it seems that it's not presenting toxicity, which is already a very good first point. And then that there would be a decrease in the impact of DUX4, so it cannot suppress completely production of the toxic protein, but they reduce about 50% the amount. And the muscles seem to be recovering, and the patients were. So there are some objective measures of muscle strength, and like the movements the patient can make that look very make us very optimistic about what this drug is doing. But there are also testimonies of patients who say, that's fantastic, now that I'm with that drug in the trial, I can start again playing with my children, which I could not do earlier because my muscles were diluted. And this type of testimony is something fantastic, something that all researchers and clinicians are dreaming of. So it's a good start. So probably it's not going to be the best drug, but at least it's opening the door for other drugs which are in the pipeline. And one important thing is that this study has identified what is named biomarkers. So something that you can measure in the blood, and that is changing in a special way in patients who are not treated. Let's say, for instance, they have a marker that the amount is increasing in the blood with time, severity increase for the disease. And if you give the drug, you see that this marker is not increasing anymore. It's first stabilized and then it goes down. So that means that indeed you are hitting with that drug the part that is destroying the muscles and certainly interfering with it. So now the field some patients mentioned was that if the company is the only one to present a drug for a disease, it will be able to sell it for really high prices. And so what is very important for patients is that several companies be producing drugs, and they are coming. The 3 more will be clinical trial this year, and they will produce drugs so that there will be a competition on the market, and the companies will have a tendency to decrease their price so that patients will go for them, not to the other one. And that's, of course, very important for patients as well. So I have to tell you 2 steps. So the first thing, you need a gene, complete gene with revealing the part coding for the protein and then the end of the gene in order to develop the disease. And this gene is producing a protein named DUX4, DUX4, and which is extremely toxic for muscles. So the protein is very useful for embryo development, but then it has to stop being produced. And in patients with FSHD, there is some screw up in the chromosomes, which make that the protein is getting into our muscle. At random, very few muscle fibers at a time, but each time it's expressed, it's going to kill that muscle fiber. Because the disease is starting very slowly, and with aging, more and more muscles are being destroyed, and so that finally a patient cannot move their legs or hearts in hands. So that's the genetic part. We need the recipe to make this DUX4 protein. And in addition, you have changes in what is named the epigenetic. That is, the DNA is associated with proteins on the chromosomes, and this association of DNA with proteins is named chromatin. And so people who do not have FSHD have a very tight chromatin, very compact chromatin over the DUX4 gene, so that it cannot be expressed in the cells. And people with FSHD, unfortunately, have some changes in the chromatin structures, that it is more open, and the gene is becoming accessible to the enzyme that will copy it to make the messenger RNA to then produce the protein. So you need two things to develop the disease. So from a genetic perspective, you need the full gene. And then from an epigenetic perspective, you need to have open chromatin. Now, genetics can again be involved in this epigenetic. That is, chromatin is made of proteins, which are expressed by genes. So you can have variations in these genes from one individual to the other, and that can change the structure of chromatin. For instance, there is one gene which has a complex name, SMCHD1. So I need to smoosh it. It's easier. So this smoosh gene is making a protein that is helping to make a compact chromatin. And when there's a mutation in that gene that destroys the protein, so it cannot make tight chromatin anymore. So the chromatin is open, and the individual with mutation in SMUT will have the last 2 gene expressed very much. So the progress in the last years has been to identify other genes that produce proteins necessary to make the chromatin, also besides SMUT. And so up to now, I think we have like 30 different ones which have been identified. And so that can be a little difference among those genes from one patient to the other. That could explain why patients with the same genetic defect are more severe or less severe affected.